Our protocol enables to measure detection and discrimination of volatile odors with a panel of odorant receptors. This method opens up the possibility to develop odorant receptor-based biosensors.
This technique allows real-time in vitro monitoring of odorant receptor activation upon odorant molecules in vapor phase. It fills gap between in vitro and in vivo approaches.
This method can be used to understand the kinetics of events that lead to odor perception, including the role of mucosal metabolic enzymes.
To begin, prepare M10 and M10-PSF according to the manuscript. Cells are cultured in 10 milliliters of M10-PSF at 37 degrees Celsius and 5% carbon dioxide. Observe the culture dish under a phase contrast microscope. When it reaches 100% confluence, split the cells to a lower concentration by aspirating the media and washing the cells gently with 10 milliliters of PBS. Then, aspirate the PBS and add 3 milliliters of Trypsin-EDTA. Let it react for approximately 1 minute until the cells dissociate from the dish. Add 5 milliliters of M10 to inactivate the trypsin, and pipe it up and down to detach the cells attached to the dish. Transfer the 8 milliliters of cell suspension from the dish to a 15 milliliter tube and centrifuge the tube at 200 times G for 5 minutes. Aspirate the supernatant and resuspend the cells in 5 milliliters of M10-PSF by pipetting up and down to break up any cell mass. Avoid creating bubbles in the tube.